and I'm really pleased to welcome Jamie Andrews to the show tonight. Jamie, thanks so much for being here. It's really great to meet you and see you.
Hi, Dr. Ruby. Thank you for having me on.
Yeah, I was very intrigued by what I saw as your postings and the information. I spoke to some other scientists who have some great regard for the work and the project and your team. So let's kind of start out maybe from the beginning. How did this team get together? What is your role? What's your interest in it? And all that, just so people have a little context for what you're going to be presenting.
Well, I started the same as everybody, really, having a big wake-up call in 2020 when the world was, thrown into this strange boiler pot situation, and we were all trying to make head nor tail of it. Here in France, obviously, we were all in a bad situation, but in France we had a vaccine passport, so myself and my family were banned because we refused to take the vaccines. We were banned from civil society. We weren't allowed to go to sports stadiums, bars, restaurants, even soft play centers for my kids. We were banished from society, as it were. And so I really wanted to start and look at the actual reasons for why we were banned from society, for not taking something that they said was necessary to not get sick.
Tonight, the intent is to lay out how he started this, what this is about, what he was trying to figure out once he learned all this. So, Jamie, I just wanted to kind of set the tone for the audience a little bit more.
Yes, yeah, that's it.
You've done a great job with how you got there and asking those questions. And I do, remember in the audience's mind, it's like, well, what the hell? Is there a virus or is there not a virus? And like I said, something made us sick, but that could be a government gain of function. So, keep that in mind as you're explaining all this. I'm fascinated to hear what you think about those things.
Yeah, that's it. I mean, back from those HIV days, like you say, there was the similar kind of dynamics between some of the main players. Kerry Mullis developed the PCR test, and really what we saw through 2020 was an entire scamdemic, I would call it, based on the results of a fraudulent test.
Sure.
The tests that we have done using independent accredited laboratories have shown that the PCR test is not testing what it claims to measure. So,
big one.
That is a big one. And that's really been our direction of travel, is to make sure that 2020 never happens again, where they lock people down, where they try and remove informed consent, where they try and remove any sort of choice to what you put in your body, even if they are, heavily coercing it. I don't want to see that for my children or anything moving into the future. So, that was really the design of the Virology Control Studies project, was to get to the end result, which is the PCR. But there's a few logical steps before you get to that PCR, which is the very first isolation. It's where you have a purified single biological particle, which they call a virus. And back during the HIV days, there was a group called the Perth Group, headed by Eleni Papadopoulos-Eliopoulos, and she inspired a guy called Stefan Lanka, who also did these control experiments. We were talking about the premise that if you take an experiment and you don't put in what you're testing, yet you get the same results, that's a control experiment. It's a legally binding part of science. In fact, it's the only legally binding part of science if you do a control. So when you're talking about vaccine studies, you're talking about a placebo. So you're talking about giving saline instead of the vaccine. So you can test whether you think that it's the vaccine doing any good. I mean, I don't think vaccines can do good, but you see its effects, right? So you take a group of people and you give one group a placebo of saline and you give another group the test subject. Well, what we're doing in these, working with these laboratories, is essentially the same thing, but this all takes place in a Petri dish. So when they in vivo, in vitro.
In
Sorry,
vitro, yeah.
when the virologists look to isolate, they can't just take a virus direct from somebody. So if you have flu symptoms, they can't just take your sputum and then go, here's the virus. Apparently, they can't do that. They can do that with bacteriology, funnily enough, because bacteria exist. You can see them.
And they're
you can
large.
see them.
They're larger than viruses
They're larger.
claim to be. Correct. So you need some exudate,
Smaller.
right? You need some exudate or something from blood. Yeah, I know that's where you're
That's
going.
right.
With it.
I mean, they say that you have to culture it. So they say that you have to use cells. So unfortunately, they cut them out of all sorts of places and they freeze them. These cell banks are clinical banks of cells, and they come from monkeys, unfortunately, from because scientists use all of these horrendously unethical things like aborted human fetuses, which are also in the vaccines when they're developing them, by the way.
Yes.
That they're using these cell lines from the kidneys of monkeys, from the kidneys of dogs and hamsters, and unfortunately people and fetuses. And they take these cell lines and they put a sample in, and they say they grow these cells out. And when a sample gets put in, they say, okay, well, these cells are dying because of a virus within that sample. And what Stefan Lanka did originally, and a few other people, and in fact, the very scientist who developed this idea of cell culture, John Enders in 1954, he did the same thing. He didn't put a virus in, what's known as control. So he took the cells, he took all of the ingredients involved in the cells, and he didn't add a virus, but yet the cells still died. And that's kind of a kicker, because to any logically minded person, to any scientifically minded person, you should say, well, if the effect happens, regardless of whether you put the virus in or you don't, then you're not showing that you have a virus, right?
Well, you're not. You can't attribute it to what you're calling. No,
you
virus,
can't. You can't attribute it to a virus. And I explain it this way: if you took a banana in a bowl and you said, I'm going to put some spit on the banana and go away and come back in 3 days, if the banana has gone black, that means that there's a virus. Well, we all know that if you didn't put any spit on it and you came back and the banana was black, it would be because it's just rotting fruit.
Right? Oxidized, right. Rotting.
So,
right.
And it sounds absurd. It sounds absurd to a layman, but that truly is what's happening. And we have shown this with independent, blinded laboratories, because again, I have no background in this whatsoever, but I have been running a scientific project and an experiment, and this is what I want to enthuse to people. We're in a world where we've got lots of information coming at us. We don't know what's up, what's down, what to trust and who to trust anymore. And you get these peer-reviewed papers coming from you left, right, and center, and health is important. It's probably the most important thing, right? And so I really wanted to enthuse being able, and it is perfectly possible because I have done it, to reach out and conduct your own science.
But it's going to be a great way to substantiate what Dr. Mullis, the inventor, said. He pretty much explained it, but I thought you just did a little more clear version of it. But he basically said you can find basically anything in there, and you can find multiples of things, but it's not going to tell you what you're sick from, or that you're sick with a flu or that kind of thing. So that's going to be very helpful in the work that you're doing. The other thing is, I don't know if you wanted to share there was one slide that we talked about.
Yeah. So if you get the slide up on the screen and
Sure.
you can have a look at the particle that we found. So this for me is the real kind of visceral part to the experiments is actually looking at microscopy. We were talking about the fact that you can see bacteria with a light microscope, but with a virus, they say that it's nanometers in scale. So it's a very, very tiny thing that you coincidentally can only see with a transmission electron microscope. Very, very powerful microscope, and you have to kill the cells. So you have to kill and fix them in a pretty noxious substance to be able to image them. And so any of the claims that you can see viruses hijacking a cell by proxy have to be an imagination. You cannot see with a microscope a virus particle size moving into a cell because you have to kill them. It has to be a stationary photograph.
I see. Okay.
So in our experiments, we have taken clinical cell lines that we were talking about before, and these are guaranteed sterile. So these are guaranteed when you buy them from an accredited bank called the ATCC. They are given to you as this is a clean sample. We've guaranteed that there's no viruses in, there's no contamination, there's no nothing in these. These are used in all clinical research. And so we took those and we did the cell culture isolation, but with no added sample to it. And then we took microscope pictures of them. And funnily enough, what did we find? Well, we found the exact particles that look like SARS-CoV-2, that look like HIV, and that look like measles.
Okay, by what metric? But what by what? By the cartoons? I don't get
so
it.
The metric that the CDC and the NIH give of what these exact particles look like. So if you look at the image on the top,
okay.
we'll start on the bottom. The bottom is what the CDC claim is SARS-CoV-2.
Okay.
So here you can see that it's round, it has these inclusions, and the size. The size is a big giveaway because it's a very, very specific size. So it's 60 to 140 nanometers in size. And in the culture at the top is our pictures that we conducted with a microscopy lab. And there we put an arrow towards an identical particle. So it's round. It has all of those kind of protein inclusions. It has the coat around the outside, and we've put the bar graph up against it, and we have an exact size. So that's probably about 100 nanometers, which is right in the middle of where a SARS-CoV-2 particle should be. And we also have the images of HIV. We have the images of measles. And actually, in our latest laboratory experiments, we've found maybe 5 or 6 more varieties. Ebola we found, which is a very kind of stringy, kind of almost snake-like particle that is just the cell lines breaking down. The cells break down, and it's more like a Rorschach graph. I don't know if you know the Rorschach, where it's like an ink blot test.
Yeah. I must interject with a couple of questions. They might be stupid.
Yeah, please.
But could the Ebola with the stringy, I mean, there's a whole issue around parasites and the role that they play, that they are the viruses. Number one. Number two, I don't want to confuse the audience. On the upper picture here, it has that disc and you've labeled it SARS-CoV-2, but that doesn't exist. You're saying that's what they claim it was, and you proved...
That's what they claim. So in the top, just to make perfectly sure, in the top, it is not possible for a virus to be in that top image.
But they claimed it was.
It's not possible. It's literally, according to scientists in clinical research, it's not possible for a virus to be in there. Okay. Yet we've found the particles that look identical to what they're claiming are viruses. So what they're doing is that they have these cells, and the cells break down, and the cells have a lipid outer layer, a kind of fat outer layer. And when they die, all of the interior of the cell line just blobs out, a bit like a lava lamp. It goes...
Okay.
And a virologist will point at the little blobs on the outside, and they'll go, oh, that's SARS-CoV-2. Oh, that's measles. Oh, That's HIV.
Right.
And so we're showing that actually they're just misinterpreting and mislabeling what is cellular debris. That's all it is. The whole thing is
They're
cellular
imposters.
debris.
Jamie, you're saying they're cellular imposters.
That's right.
Got a new phrase. They're cell imposters. Well, they've done a lot of cartoons, right? The colored cartoons with the
That's
spikes
right.
and all that. This is
That's right.
what you're showing is at the nanometer level, the fraud.
Exactly. And this is the problem is that
Wow.
most people think that they don't have they don't have access to this type of equipment. And so, what I'm saying is twofold is that actually you do have access to this equipment. You've just got to know, and you've got to be able to be enthralled and and motivated to do it.
Yeah.
But apart from that, you do have access to be able to check and verify what these people are saying, because I want you to check and verify what I'm saying too.
Mm-hmm.
And actually in our latest experiments, I work with a guy called Albert Matthews who said to me, I've seen your experiments and I think that they're great and I think that I believe them, but honestly, I want to see for myself too. And
yes.
All respect to him, because he went out and did exactly the same thing. And we found even more. We verified our results and we found the same. We found all of these types of things. So, I think that it's incumbent on people to look, but, this was the problem that we came up against, is the fact that because it's on such a nanometer scale, because it's on, it's invisible to the layman, it's invisible to the general public. You can't just take a microscope off the shelf. You can't go to Walmart and just buy a microscope and see these things because they're claiming that they're too small. Mm-hmm. You have to go through all of these laborious, things to be able to zoom in and go, oh, well, actually, they're talking crap.
Mm-hmm. Actually, that's, and they know that. They know that.
Yes,
that's why viruses have given the powers that be so much ability to dupe and fraud and murder and maim. So, Jamie, we're going to wrap up for this session. I'm so glad I had you on. I knew the minute I started to see your work, and I didn't even understand all of it. I understand a lot more of it now that you've kind of walked us through it. It's fascinating. I want to make sure that people can get to your work, can get to because you are trying to get it out on social media. You tell us where to find you, Substack, X, and we'll put it on the screen and people can find you. I'll also put it in the links.
All of the results that we have are being released open source, so you can take those results and use them as yourself. You don't even need to kind of quote me or need to do anything with them. Please repackage them, take them, and do what you will. There are lots of people out there that are submitting them to local courts and local jurisdictions, challenging people with these results. So going and saying, look, can you show me evidence that you've based, local communities where you can take them in. So that's all being released open source on Substack, which is substack.com/control studies. And then I'm also on Twitter. You can just ask me any question about these. I'll be happy to talk to you a little bit more in detail about them. I'm at Jamie. I'm at Jamie A again. Okay. Because I've had so many accounts banned on there through shock horror. I think I'm on my kind of fourth or fifth iteration on there. But
wow, so you're not just a Johnny come lately. You've been at this for a while, and they've been holding you down. And we have the Twitter handle and the Substack on the screen. In fact, we'll make sure it's on the screen throughout the entire show.
Thank you.
For later, but I wanted to make sure we mentioned that people can. And I wanted to add just one more point. I really want to let the audience needs to understand that real science is not afraid to be replicated. And not only are you're encouraging it, you're sharing everything so that others can replicate it, and that's what's been missing. In this whole fraud.
No, thank you. No, it has. It's been out for the initial experiments have been out for 18 months now, and I have put a call to action for virologists that want to, show that we're wrong to please do it. And it's very easy for them. It's cost in the region of $25,000 to put these few experiments together. But for a virologist, it's like reaching into the store cupboard. For
Okay.
them, if they have a laboratory, it's like reaching in and pulling out a stapler and a pencil and a
Sure.
pad of paper, and they can do it in a couple of days. But funnily enough, they refuse to do it and show that we're wrong. So,
Self-serving.
there has to come a point where, maybe we're onto something. But
Of
yeah, as
course.
you say, we put everything on the table, and we're open to being showed that we're wrong, and we're open to being verified or falsified.
And see, that's what's been missing. And anyone who refuses or turns you down or scoffs at replicating it is really self-serving. They're trying to preserve their paycheck. And once again, it's always the paycheck.